علي.
بسم الله الرحمن الرحيم. أنا نشوى محمود Laboratory Analyst في الواتر Chemistry Department CPHL Master Holder of Environmental Science عين شمس University. النهارده خلونا نتكلم على حاجة كلنا بناكلها، أغلب الناس اللي عايشين في القاهرة أو مناطق غير ساحلية بيكونسوم النايل تيلابيا أو الأوريوكروموس نايلوتيكس أو النايل بولتي فيش. غالبا يعني كلنا بناكله like once a week، والناس اللي بتحب السمك شوية بتاكله like twice a week. So let's have a look. Is it poisonous? Is it not? First خلوني أبدأ بالgood side, bright side. What are the good things about consuming fish? Tilapia contains omega-3 fatty acid, which the human body can only obtain through consumption of certain foods. The surprise is the tilapia fish has high content of omega-3 fatty acid than other meats. يعني اللي موجود في السمك من الأوميجا 3 مش موجود في الchicken، مش موجود في البيف. Also, الأوميجا 3 fatty acid is very important للbrain growth، ودي أكتر حاجة هنتكلم عنها دلوقتي. The effect of methyl mercury found in fish according to the brain. الأول خلونا نشوف طب الميثايل ميركوري ده أصلاً جالنا إزاي؟ دخل السمك إزاي؟ First الميركوري هو ثالث dangerous environmental toxin موجود على الأرض بعد الليد والأرزنك. بيوصل للenvironment through شوية practices by humans, mining, recycling facilities, medical wastes, and cement, a lot of things in the industrial consumption. وبرده في sources للفود، واكتر سورس مشهور هو الهاي فركتوز كورن سيرب، وكمان الرايس في بعض البلدان اللي بيكون فيها يوز للميه بيكون فيها high levels of methyl mercury. طيب هنقسم المحاضره ل 2 parts. The first part هنتكلم على الميركوري في الاكواتيك لايف، وصل ازاي الميه، السايكل بتاعت الميثايليشن بتاع الميركوري، وكمان جوه الفيش بيدخل الفيش يعمل لها ايه because this is the main concern now. وبرضه هنتكلم على لما بيوصل بقى للهيومن الهيلث افكت عامل ازاي و how to prevent it. Let me start first with mercury in the aquatic life. Mercury بيبدأ الأول بينزل المية through الwaste whatever the source of the waste. أول ما بيوصل للمية بيحصل فيه ligand combination with organic materials. After the ligand combination, methylation occurs by microorganisms. And then methyl mercury is dispersed through the consumption بتاع الفيش أو بتاع all the organisms present in this water. The concentration بتاع total mercury والmethyl mercury for most of the food is not dangerous, is not of concern. The only food that has concern of the methyl mercury is the food coming from the aquatic environment. In this research, احنا شغالين على البولتي fish, but in general, the aquatic food is having the highest source of methyl mercury بيتعرض له الإنسان. في أي فود تاني الموضوع بيبقى قليل جدا up to less than 0.1 microgram per gram. حاجة ثانية actually 90% of the mercury that exists في كل الفود around us is in the form of methyl mercury. That's why methyl mercury is our area of concern for now. This is the mercury cycle we're having. First, the mercury is emitted to the atmosphere. After that, the mercury will go in the rain and snow and as gases and particles. Then the mercury accumulates in lakes, reservoirs, and forests. Finally, is transported through water from one place to another and having the organisms. The last part, methyl mercury bioaccumulation in the food web. That means in the mercury مش بينزل المية methyl mercury, it is methylated في المية through some microorganisms present there. طيب كده الميركوري وصل المية والسمك خد الميثايل ميركوري. طب ايه اللي هيحصل بقى في السمك لما خد الميثايل ميركوري؟ اولا الميركوري هيوقف الجروث او الديفلوبمنت بتاع اليونيسيلولار اورجانيزمز الموجوده في الميه like the algae, the bacteria and the fungi, as well as some small fish. طيب ده هيسبب ان البيج فيش اللي هو من ضمنهم النايل تيلابيا will not be able to find enough food for consumption. ده طبعا هيأثر على the number of embryo and the larval fatalities. mainly لما الميثايل mercury بيوصل للفش الفش بيجت affected through the brain, the placenta, and actually the muscles of the fish اللي احنا بنكونسوم. دي السايكل اللي بتحصل للاوكسيديشن of the methyl mercury. First, mercury comes through the air. Some of the mercury goes for the oxidation أو إنه يحصل يبقى mercury valence, divalent, and then بيوصل للمية يحصل له methylation by microorganisms, and then this methylation بيخليها more stable to be consumption, to have the consumption through microorganisms or through the fish. Okay, كده خلصنا البارت بتاع الفيش, how it's affected by the presence of methyl mercury. Let's get back to the part of the human. What happens after ingestion of aquatic food containing methyl mercury? When people ingest methyl mercury contaminated food, methyl mercury is separated in the gastric acid. It combines with the cysteine among the amino acids in the duodenum, and almost 100% of the methyl mercury absorbed goes to the blood. Afterwards, it combines with the hemoglobin of red blood cells through the portal vein, and it causes the disorders of the neurons. في research اتعملت على animal experiments بتقول إن 95% of the mercury can be absorbed by inhalation. Although مش دايما بنتعرض لمشاكل في الميثايل mercury by inhalation, بس هي برضو considerable amount to be researched further, to go under further research. بس خلينا نقول إن هو mainly it's easily dissolved in fats, lipophilic, وده معناه إن هو بيشو 17 to 35 times faster than inorganic mercury. The maximum blood methyl mercury concentration was found 6 hours after food exposure. يعني once we eat food, if we took blood samples from a human, according to this study, 6 hours after food exposure, we will find that 95% of the intake was absorbed through the blood. It is known that when people are exposed to methyl mercury, blood mercury moves to the follicle in the hair growth stage, وعشان كده أغلب الناس اللي بيبقى عندهم mercury acute toxicity, they have a grayish hair and is accumulated there. It has also been shown that the mercury concentration in hair is proportional to the blood concentration, وعشان كده some of the human researches are done not to the blood but to the hair because they are proportional and having exactly the same percentage. It is known that methyl mercury is converted to bivalent inorganic mercury and undergoes oxidation and reduction, as we have seen in the picture. Methyl mercury releases oxygen radicals at decomposition. ده معناه إن الميثايل mercury, once it's got inside the body, we shouldn't be so happy that it's going out for excretion, because to get away from the methyl mercury form, to be able to go excreted, we have the ROS, reactive oxygen species, which acts as toxicity, general toxicity. We'll see now the effect of these ROS. It causes severe damage to cells by activating the chain of lipid peroxidation, as it has high fat solubility. فأكيد هيبقى أول حاجة هنأتاك blood-brain barrier, passing the blood-brain barrier. Here is what happens when methyl mercury goes under decomposition and reactive oxygen species are released. Number one, irreversibly inhibit the selenoenzymes. Selenoenzymes, the use of an example, the glutathione. The glutathione found in the brain, it helps the movement or the transportation of the neurotransmission through the brain. After that, the oxidative damage that happens to the cell. After that, reduction in natural killer cell activity and disruption of DNA repair with the interfere with DNA transcription and protein synthesis. Finally, it causes the disruption of the mitochondrial membrane. Let's get a closer look to the effect we're having. First, we're having DNA damage and we're having excitotoxicity, then cell death, calcium imbalance, neurogenesis impairment, neuroinflammation, neurotransmission alterations, and oxidative stress. I'm not going into details with the clinical side because this presentation is mainly focusing with the fish toxicity in human and the study that we had on some regions in Egypt, getting samples of fish and analyzing it. And of course, that recommends further research for human investigations. Mechanism of toxicity in human. First, methyl mercury has high affinity with the sulfhydryl protein group, When it is ingested into the human body, it combines with glutathione to make methyl mercury glutathione compound. And this is the compound that has the easy access through the blood-brain barrier going back and forth. It easily crosses the blood-brain barrier and the placenta. Placenta here means that we are having an effect on the fetus and newborn babies. And here, the concentration of the mercury, 90% of the mercury, as we have said, the methyl mercury actually, is distributed in red blood cells. This is the picture that explains how it's affected the transportation of the neurotransmission. The blood-brain barrier is having actually two gates. The first one is called the LAT, L-type large neutral amino acid transporter, and we're having the multi-drug resistant protein MRP1. What happens? Methyl mercury in blood. Methyl mercury goes inside through the blood-brain barrier, but carried on the L-cysteine amino acid. It goes inside the brain. In the brain, we're having glutathione. The glutathione carries the methyl mercury again, actually multiplies it, and then goes back through the MRP1. So this means that if we had less amount of glutathione, we can control the going back and forth of the methyl mercury through the blood-brain barrier. That's why the acetylcysteine has a great role in this, because more production and more presence of acetylcysteine means more glutathione, means more of the cycle. Neurotransmission alterations, it's the same idea we're speaking about. It's how the methyl mercury carried on the sulfhydryl group goes back and forth through the blood-brain barrier and through the cerebrospinal fluid barrier inside the brain, affecting the neurotransmission. This is a closer look. If we are having the methyl mercury in the brain, on the L-cysteine in the blood, and on the glutathione inside the brain, this means that the neurotransmission moving back and forth through the blood and through the brain, it will have some great amount of latency because the gates of the passing is very busy carrying the methyl mercury. Okay, here is the excretion. As long as we said that it's lipophilic, so the excretion will be through the feces and through the bile. Methyl mercury is excreted in the bile, but part of it is reabsorbed through the enterohepatic cycling and flows to the liver. It can't be stopped. Actually, even if it's going to get excreted by the bile, it will not get out. It will go away again with the enterohepatic cycle. So the only way to get into the methyl mercury is to prevent its consumption from the very beginning. Most methyl mercury is dissolved by methylation and excreted to the feces in the ion form. The feces occupied 90% of total mercury in the excretion, and that's a good idea. That's good news, because that means that we are not so much depending on the bile and the enterohepatic cycle. Good information to know that once you get consuming methyl mercury, you're having 70 days half-life time to get rid of the methyl mercury absorbed. This is a wrap-up of the infection we're having. Actually, we're having of the contamination and the toxicity we're having, actually, we're having toxicity and contamination through affecting a lot of the organs in the body, because it moves with the blood as on the L-cysteine, and it goes in the brain and the blood-brain barrier. It affects the placenta, so it causes developmental delay, embryonic malformation, and dysplasia motor disorder, causing the very famous Minamata disease that happened in Japan. And lastly, affecting the neurotransmission and the neurological dysfunction, proliferation disorder, differentiation disorder. Okay, after that, this is international, and this is some facts about methyl mercury. Let's see what happens in the Nile River. Nile River has 124 point source of waste. This is according to the National Water Research in 2000, and 67 of it is agricultural drains. They found that the metal content in winter in the fish mussel is higher than in summer, and that's due to the flow of rain that leads to increasing the wastes in the water. The bioavailability of metals may be influenced by physiological activity of fish during different seasons. Different seasons depend conditions such as salinity, pH, and hardness. Depending on the previous stated facts, This was the study. The study was made on Lake El-Burulus in Kafr al-Sheikh, Nile River in Qalyubiyya, fish farms in Beni Suef Governorate, and Nile River in Governorate. The study was, we had samples of the fish in the beginning of every month of the year through 2 years, 2017 and 2018. And we analyzed these fish having total mercury, and then methyl mercury was calculated from that measurement we had using the ICP conductive. And actually, the results were compared to the US FDA and the US EPA. That was the results of 2017 for the 4 governorates we're having. As you see in the curve, we're having 0.3. This is the head of the curve. And also we're having that the maximum allowable limits is 0.50 microgram per kilogram, which is good news. It means that we are not even close to the higher level of methyl mercury. These measurements were made in fish, not in human blood. And this is the results of 2018, which is the highest level of the graph is 0.4, and we're also still beyond the maximum allowable limits allowed by the EPA and allowed by the FDA. So according to the numbers here, we're not reaching 0.5, we're not reaching 1 microgram. So the conclusion that in Egypt, the fish species did not exceed the maximum allowable limits assigned by the FDA and assigned by the EPA. And that means that the probability and the frequency of acute and mercury poisoning in Egypt is not here. Here comes the question. So you mean that fish in Egypt is completely safe and not toxic? Actually, no. The answer comes here. The answer is in these 2 words, bioaccumulation and biomagnification. Bioaccumulation means it's the increase in concentration of a pollutant in an organism. That means that when somebody eats food every day, the accumulation of the very small concentrations found will cause big problems or poisoning. And also the biomagnification, it means changing the consumer. People eating fish, the fish having 0.1, for example, in human, it can be multiplied like times 10 power 6, which is a considerable amount. That's why further human investigation is recommended by this study. Bioaccumulation, biomagnification, that's further explanation for this using the graph. And finally, these are some international papers and researches that were made on the methyl mercury found in fish and published globally. Number 1, in Marine Pollution Bulletin Journal, biomagnification of methyl mercury in a marine food web in Lezo Bay, North China, and associated potential risk to public health. Actually, they found that the fish itself, one fish or the consumption is not causing acute toxicity or poisoning, but they found that biomagnification and bioaccumulation, yes, can cause very high potential risk. The second paper we're having today, health risk assessment of methyl mercury from fish consumption in a sample of adult Qatari residents. In Qatar, most of the people, Qatari or not Qatari, they consume high amounts of fish, and it affects within the permissible levels set by the European regulations, European Union regulations. And they found that the fish itself is not having a problem, but the high level of consumptions of Qatari people to fish, depending on it as main source of protein, that's how it is affected. The last paper we're having today, the causes and effects of mercury and methyl mercury contamination in the marine environment. That was a review that said that increasing the mercury level in fish species and causing the adverse health risk to humans through the biomagnification, it was highlighting that methyl mercury around the world is not an issue of speaking as acute poisoning or acute toxicity, but it is something that should be managed by biomagnification and bioaccumulation. Finally, ناكل سمك ولا ما ناكلش، حلو ولا وحش. This is the take-home message. We said that mercury-containing fish بيبقى شكله whitish skin بتاعه بيبقى أبيض مش بيبقى gray, and الeyes بتبقى grayish. فاحنا دايما واحنا بندور واحنا بنشتري سمك هنجو for البينك والريد color مع احترامي للنيترايت يا حنان. And then Nile tilapia fish is safe to eat up to 2 servings per week. Each is 200 grams according to the US FDA and the FAO. And that's it for today. For further inquiries, questions, and discussions, please feel free to contact me via email. Thank you very much.
thank you, our three scientists for the third session. And I think they made our food habit very dark. Meat is contaminated with nitrate. Fish is contaminated with mercury. And potato and coffee is contaminated with acrylamide. I think they're pushing us to go for a vegetarian habits. I don't know what are we going to eat. It's very dark now. I don't know. Are we going to eat now, Dr. Nancy? I mean, we have to be very careful for selecting our food now. We open the session for any questions, please. Any questions? Okay.
Good afternoon, everybody. I'm Dr. Hiba Sharafuddin, Professor of Clinical Pathology. معلش عايز صوتك جدا. I'd like to ask Dr. Nashwa about the first method of assay of mercury. I would like to ask Dr. Nashwa about method of assay of mercury in fish and in human blood. Second, is this related only to poultry fish or other kinds of fishes in the Nile River? And third, in countries like Japan, which is the main feeding habit there is eating fish, maybe 5 times per week. Is this problem is prominent there? Because eating fish in Egypt is not so frequent. And regarding poultry, it is the cheapest type of fish in the market, which all the population can eat now. Is this also can apply to other types of fishes like bori or whatever, salmon or whatever, tuna? 3 questions. Thank you.
Doctor, according to your question, the question can هل الكلام ده بيجو for other fish, especially in Japan, species that they are consuming because it's the main food there? Actually, yes. هو in general في كده general rule according لان احنا now لما كنا نتكلم على البايو magnification البايو accumulation ان الميركوري او الميثايل ميركوري concentration ما بيبقاش عالي في الانفايرمنت، هو بيبقى تراكمي. فده means في type of species في جابان اللي هي تبقى السبيشيز الصغيره اللي عمرها قصير بيبقى الكونسومشن بتاعها every day مش of a considerable amount او considerable concentration او effect on humans. بس في species هناك اللي هي species الكبيره mainly things like sharks like very big fish that consumes other fish. فده معناه ان انا بزود في الفود ويب البايو ماجنيفيكيشن، يعني احنا هنا بناكل سمك بيتغذى على tiny organisms و plants. في جابان لا، في جابان هم بياكلوا سمك بيتغذى على سمك تاني بيتغذى على سمك تالت، فهم عندهم 2 risks. الريسك بتاع البايو accumulation جوه البني ادمين بسبب الاماونت اوف consumption number of servings per week، وبسبب ان هم بياكلوا البيج فيشز اللي بيكون حاصل فيها already بايو ماجنيفيكيشن، فهم مش بيستهلكوا فيشز بيكون فيها اصلا الميثايل ميركوري of low concentration. That's why لما جت حصلت الكرايسيس بتاعت الميناماتا disease it happened actually in Japan. يعني malformations Minamata disease, this is the malformations that happen for the newborn and the placental deformations بتاعت الفيتوس. هي لما حصلت في جابان أكتر حاجة. ليه؟ علشان healthy practices بتاعتهم through consumption و through الفيش ده اللي كان سبب عندهم يعني. that's why الريسيرشز اتعملت على other species والنهارده بس we were focusing on us on our one health approach in Egypt, but yeah there are way too many research and the rule is one for all of them: bioaccumulation and biomagnification. Thank you very much. في الhuman I will not speak about it because I am yes the research was made on fish and I am a pharmacist working in water chemistry laboratory CPHL and my master's was environmental science detecting of fish, so I will not go for a human part. This is clinical part. I will go for using. We use the ICP inductive coupled plasma, and it was a device. The process, I can send you the SOP or the paper that we followed, but mainly it depended on the complete ignition of the muscles of the fish, and then the nitrogenation of the muscles, and then going in the device, and the ICP mass gave us what we need. The results were only for mercury. Yani these measurements were for mercury, and then we added the factor 1.704. That factor was to calculate the methyl mercury from the mercury, because in Egypt it's not easy to calculate methyl mercury directly. We're not having that very advanced devices to go for these minute nanograms concentration. Further questions?
في أي أسئلة أخرى؟ محمد، في الأستاذ عاوز أو الدكتور عاوز يسأل.
أستاذ حضرتك يبقى last question.
ماشي.
My question for Dr. Nashwa, please. From the lecture, I want to ask the fish farms is more safe than the Nile fish, because there is no exposure to the environmental mercury and
yes, that's a very important question. Thank you.
The second one, sometimes also in some types of tuna come from Vietnam and from Thailand and from other countries. There is also concentration of mercury, and some recommendations come to avoid specific types of tuna.
Okay. طيب, let me go first for your first question. The first question was about fish farms. هو بس خليني اوضح مفهوم الفيش فارمز عموما. هو الفيش فارمز بتبقى مش filled مثلا من water حصل لها treatment. Mainly the water used in the fish farms يا اما بتبقى coming from a near water source, يا اما بتبقى water prepared for fish development or for fish growth. يعني اللي احنا سحبنا منهم السامبلز دي، عشان كده انا مش هعرف ارد على الكويستشن بتاع حضرتك قوي، كانت نايل فيش فارمز. يعني كانوا ناس ساكنين في هذه المحافظات على الشط، وهم عملوا زي شباك لاستزراع السمك. مش خدوا يعني مثلا في مثلا في الاسماعيليه وفي الانطره، لان هم بعيد عن كل سورسز اوف ووتر، بيعملوا هناك الفيش فارمز تailored ووتر زي ما حضرتك بتقول كده، بتبقى فعلا ما فيهاش البوليوشن الجنرال بوليوشن. بس الفيش فارمز بتبقى موجوده على النايل ما بتبقاش تailored ووتر، بتبقى هي الووتر نايل، بيبقى السبب ايكونوميكال مينلي، يعني ان يحصل استزراع للسمك وزياده كميته والحفاظ عليه ده بس. بالنسبه للسؤال الثاني حضرتك سالته regarding التونه او الحاجات اللي جايه مستورده، هو عموما احنا ما عندناش ميثود اوف بروتكشن من الميركوري غير ان احنا نقلل الكونسومشن بتاعه، سواء كانت التونه دي ايجيبشن تونه which is ما فيش، او تونه جايه من بره. that means ان احنا لما هناخد التونه مش هنعمل لها consumption once a week as long as ان احنا عارفين ان هي مثلا جايه من فيتنام، جايه من جابان، جايه عموما من اسيا، فاهمين بقى ان هناك عندهم high pollution و high consumption. ولو ان برضو انا I didn't have research on this هم فعلا عندهم high pollution و high consumption ولا لا، بس عموما الجنرال recommendations للتونه بسبب الباكجينج بتاعها بيبقى فيها levels of mercury بسبب الباكجينج مش بسبب الفيش اعلى من الفيش الفريش فيش عموما، فهيبقى recommended serving ليها once per week 200 grams او هتبقى every 2 weeks. That's the question I'm having. Any further questions?
دكتور محمد قال إن ده آخر سؤال عشان الوقت، والحقيقة في النهاية أنا يعني بشكر تاني الدكتورة نانسي على المؤتمر الرائع ده، وإن هو كل سنة بيبقى أكثر يعني غني أكتر من اللي قبله. وبشكر المعامل سواء المركزية أو الفرعية، والواحد سعيد إن كل هؤلاء الزملاء والأبناء ليا وإخواتي اللي بفتخر بيهم، والدور الهام اللي بتقوم به المعامل والرقابة على الأغذية في الوزارة، الدور الرقابي والدور التوعوي، لأن طبعا المحاضرات اللي اتقالت دي فيها معلومات رائعة
ومفيدة للجميع سواء للمصنعين أو للكونسيومرز. يعني هو المعامل تعتبر الجندي المجهول ولا وراء الحفاظ على سلامة وصلاحية المواد الغذائية ومياه الشرب. شكرا لحضراتكم جميعا وعقبال كل سنة إن شاء الله.
استاذن حضراتكم، احنا عندنا مكانين لللانش دلوقتي، مكان قاعة السرايا اللي قدام خالص ومكان على اليمين، عشان ما نعملش تزاحم في الخانتين، فالمكانين ان شاء الله يكفونا وزياده. ان شاء الله مش هناكلكم سمك ومش هنشربكم قهوه غامقه ومش هناكلكم عيش ولا بطاطس ولا لحمه معلبه، وربنا يستر.